The leading theme in the session this morning related to the enormous, fascinating molecular heterogeneity of AML. We are beginning to understand that AML is a very complex disease. Complex in the sense of the different genetic abnormalities that the disease acquires during its origination, but it's also complex in terms of sub-clonal structure. And the various talks this morning addressed this question, and new data were provided. Data about how from an early, let us say, pre-leukemic condition in various steps, the disease, full-blown AML emerges. Also data were presented about what happens after treatment. After treatment, are these early changes gone? Do they disappear? Or do they persist? And do they perhaps predict outcome of treatment? Data were provided also about different disease conditions, acute myeloid leukemia, myelodysplastic syndrome, and related conditions like aplastic anemia. It was an exciting session. There was a lot of discussion, and the enormous interest in this new knowledge relates to the fact that we hope we will be able to better treat these patients and take into account the differences among patients, the diversity, and personalize the treatment choice according to these differences. Another, I think, potential implication is that hopefully someday we will be able to intervene in this development process and prevent the development of leukemia. We are not yet there, but the data presented today all related to this topic. A new area that is being discovered is that there is much more inherited leukemia than we had ever thought. So about 10 years ago, the prevailing view was that acute myeloid leukemia is an acquired disease with no hereditary factors. But with the ability to find gene mutations, both in the leukemic cells and in the germ-line, a lot of hereditary components, inherited leukemias, familiar predisposition syndromes are being discovered. And this is very important because these patients are predisposed to develop leukemia, or their family members are predisposed to leukemia. And it leads to different management approaches in these patients. Another presentation dealt with the question of chemotherapy resistance, and in particular cytarabine resistance. Cytarabine is one of the cornerstone drugs in the treatment of AML for a long time, and a new mechanism of resistance was presented. This was interesting. This was entirely novel data. Is not yet published. It is online now. And it opens avenues for perhaps intervening so that resistance to this important drug can be circumvented. That was also very interesting data. I think everyone was excited to see those data for the first time this morning.